I am Atsushi Okumura, a veterinarian at National Institute of Allergy and Infectious Diseases. I thank the organizer for giving me this opportunity. First, let me talk about One Health. As shown here, One Health means that the health of humans, animals, and environment is all interrelated as one. Namely, in considering zoonosis, we must not think only about humans, but human, domestic animal, and wildlife health must be seen as one. We must understand that air, water, and soil are produced and shared by all forms of life on the earth that live on one world and must seek for the health of them. In Japan, Memorandum Fukuoka was signed in 2016 at the Second World Conference in Fukuoka. It was agreed to enhance the collaborations between the veterinarians and physicians for the prevention of zoonosis. One Health is also discussed in the United States, where veterinary universities provide education focusing on One Health. When faced with an injured animal, veterinarians should not only treat the animal, but should also provide preventive care to maintain health. Veterinarians should prevent diseases that are transmitted between human and animal from the perspective of food safety and security. In toxicology and ecology, veterinarians must understand environmental diseases that impact both humans and animals to control, prevent, diagnose, and treat them. Manhattan Principles are important in discussing One Health. Health experts from around the world met in 2004 for a symposium organized by the Wildlife Conservation Society and hosted by the Rockefeller University. Using case studies on Ebola, avian influenza, prion disease, and chronic wasting disease as examples, the experts discussed approach to control threats to the health of life on Earth. The product under One Health approach is called the Manhattan Principles and lists 12 recommendations to international organizations and governments. The Manhattan Principles recommends to recognize the essential link between human, domestic, animal, and wildlife health and the threat disease poses to food supplies, economies, and the biodiversity, and to seek opportunities to fully integrate biodiversity conservation perspectives and human needs when providing solutions to infectious disease threats. To reduce the demand for and better regulate the international wildlife and the bushmeat trade, to restrict the culling of free-ranging wildlife species for disease control to situations where there is international scientific consensus that a wildlife population poses a significant threat to human health, food security, or wildlife health more broadly, and to provide support for global wildlife health surveillance network as part of early warning systems for emergence and resurgence of disease threats. In the concluding part, it reads, "Only by breaking down the barriers among agencies, individuals, and specialists can we unleash the innovation and expertise needed to meet the many serious challenges to the health of people, domestic animals, and wildlife, and to the integrity of ecosystems. Solving today's threats and tomorrow's problems cannot be accomplished by yesterday's approaches. We are in an era of One World, One Health, and we must devise adaptive, forward-looking, and multidisciplinary solutions to the challenges that undoubtedly lie ahead." Zoonosis infects the disease from animal to human. As of 2000, there are about 1,400 causative pathogens to human infectious diseases, 60% of which are animal-derived infections, of which 75% are emerging infectious disease. Not only Ebola virus and COVID-19 virus that cause fatal infections, indigenous bacteria of pets such as Campylobacter cause infection to human and recognized as zoonosis. This I borrowed from MHLW's Handbook of Zoonosis, which shows infectious disease found across the world. In Africa, there are not only Ebola hemorrhagic fever, but there are other fatal infections including Crimean Congo hemorrhagic fever and Lassa fever. Now let me talk about Ebola hemorrhagic fever, the theme of the talk, in detail. This shows virus, host, and geographical distribution known so far. In addition to Ebola, in the family Arinaviridae, there are Lassa fever, Argentine hemorrhagic fever, Bolivian hemorrhagic fever, and Venezuelan hemorrhagic fever seen in South America. In Asia and Europe, including Japan, there is hemorrhagic fever with renal syndrome caused by Hantan virus of family Bunyaviridae. There is also dengue virus of family Flaviviridae causing epidemic in Japan a few years ago. Ebola virus from the family Filoviridae has genome with single-stranded negative RNA. Marburg virus is in the same family. Today I will talk about Ebola virus. Ebola virus is mainly pathogenic to humans and primates, causing fatal infection. In Japan, it is designated as Class 1 infectious disease. The highest level, namely BSL-4 compliance, is required. The first Ebola outbreak was in 1976 in Yambuku village near Ebola River in Zaire and Sudan. The virus was named after this river. There's only one strain of Marburg identified so far, whereas 6 strains of Ebola have been isolated. 4 are from Africa, causing fatal infections to humans. They are Zaire, Sudan, Bundibugyo, and Thai forest strains. Ebola-like gene fragments were found from a bat caught in Sierra Leone in Africa in 2016. This was identified as a new species called Bombari virus. Reston causes fatal disease to monkeys, but it does not infect humans to develop disease. All monkey pandemics were seen among cynomolgus imported from the breeding facilities in the Philippines. The first outbreak was in Reston, Virginia in 1989, followed by Alice, Texas in 1990, Siena, Italy in 1992. Alice and in the Philippines in 1996 among cynomolgus. Reston strain was also found in July 2008 during the investigation of porcine infections in the Philippines, but it is still not known as to how the pig was infected. The natural host of Ebola virus has not been identified, but several fruit bats are considered likely. Transmission from monkey to human is reported, but monkeys are not carriers but final hosts like humans. Transmission route from natural host has not been identified, but main route of infection is through the contact with the blood and bodily fluid of patient. Infection from monkey to human is likely to be through bushmeat. The relationship between human pandemic and that of primate is pointed out. The first Ebola hemorrhagic fever outbreak was in Zaire and Sudan in 1976. In Sudan, 284 people were infected and the mortality was 53%. In Zaire, 318 people were infected with the mortality of 88%. Since 2000, there has been an outbreak in Central Africa almost every year. In Uganda, between 2000 and 2001, 425 people were infected, 224 died. In the Republic of Congo, between 2001 and 2002, 124 were infected and 74 died. You may remember the big outbreak between 2014 and 2016 in West Africa. Now let's look at the Ebola outbreak in West Africa in 2014. The first case of the outbreak in 2014 was reported in December 2013. An 18-month-old boy in Guinea was believed to be infected by a bat. In the region, there were 5 other fatal diarrhea. On January 24, 2014, formal medical alert was issued by the local health authority. Ebola hemorrhagic fever soon spread to Conakry, the capital of Guinea. On March 13, 2014, the National Health Authority issued an alert for the disease of unknown cause. Right after that, the Pasteur Institute in France confirmed the disease to be Ebola hemorrhagic fever caused by Zaire Ebola virus. On March 23, 2014, there were 49 confirmed cases and 29 deaths. WHO formally announced the outbreak of Ebola hemorrhagic fever. In 2014, at the beginning, because it was thought that Ebola infection in Africa was not a major public health issue, measures to control it was delayed, causing a major spread of the disease. For many years, the region suffered from political issues such as civil war and terrorism, causing uncontrolled poverty and fear that led to the delay of appropriate countermeasures. And because of local culture, there were people who did not comply with the countermeasures in place. First, there are many patients in Guinea, but then cases spread to Sierra Leone and Liberia. In Liberia and Sierra Leone, there was a rapid increase. The reason for such a rapid increase were it took time to implement countermeasures since these countries never had an Ebola outbreak in the past. Guinea, Liberia, and Sierra Leone share borders where local people moved freely across the border. And because the local people did not have much knowledge of Ebola, and since there were several areas of outbreak with broad geographical expansion, it was difficult to control the infection. If you look at the number of cases and deaths, in Sierra Leone and Liberia, located south of Guinea, the country of first outbreak, there were many cases and deaths, whereas in the north in Senegal, there was only 1 patient with no death. This is because Senegal on March 26 closed the border with Guinea and stopped the traffic to prevent the spread of infection. On July 20, Liberian American nationality traveled by air from Liberia to Nigeria and developed the disease after arrival. The positive cases and deaths in Nigeria are primary and secondary infections. The first case in Mali is a 2-year-old girl who traveled to Mali from Guinea with grandmother and developed disease. The infection spread with 8 positives and 6 deaths. And this is about infectivity. Recently, because of COVID-19, we often hear the term basic reproduction number or R naught. This is used to indicate infectivity by showing how many people can a patient infect. Basic reproduction number is the basic infectivity of virus. Effective reproduction number is the infectivity at a certain time point during outbreak. The outnote for measles is 12 to 18. 1918 pandemic strain of influenza is 2 to 3. COVID-19, although it varies between countries and regions, is between 1.4 to 2.5. As for Ebola in 2014, since the transmission was mostly through contact, the outnote in Guinea was 1.5, Liberia 1.5, and Sierra Leone slightly higher at 2.5. After the Ebola outbreak in 2014 in West Africa, there was an outbreak in Democratic Republic of the Congo in 2018 to 2019. This was the 9th outbreak in the country since 1976. In May 2018, cases were found in the remote northwestern region of Bikoro in Equator Province. 54 were infected with 33 deaths before the end of outbreak was declared 2.5 months later on July 14. Though the end of outbreak was declared on July 31st, 4 cases were confirmed in North Kivu Province. The outbreak spread to urban region with a total of 3,481 cases and 2,299 deaths. And this was the second largest outbreak after the outbreak of 2014 in West Africa. Initial symptoms of Ebola hemorrhagic fever are similar to those of influenza, namely fever, headache, arthralgia, and abdominal pain, with vomiting, diarrhea, loss of appetite, and asthenia. Dysphagia due to pharyngolaryngeal swelling, dyspnea, and myalgia are seen. With progression, hemorrhagic symptoms, including bleeding from subcutaneous region, eyes, and melena are observed. With progression in acute phase, coagulation failure leads to bleeding from mucosa and skin, as well as bloody urine and stool are observed. Neurological symptoms are also seen, and these lead to multi-organ failure and death. This is the relationship between Ebola disease progression and clinical findings. In the initial stage, there are GI symptoms, diarrhea, vomiting, fever, headache, anorexia, and asthenia, with leukopenia, thrombocytopenia, increase of ALT and AST caused by hepatic disorder, and these are followed by rash and hemorrhagic manifestations. And there are respiratory acidosis due to respiratory failure, neurological symptoms with hematological findings including the increase of BUN and creatine due to renal failure, electrolyte imbalance, metabolic acidosis caused by the increase of lactic acid, and decline of bicarbonate, myoglobinuria due to myolysis, and increase of CPK are also observed. Renal failure seems to be related to the decline of circulatory volume and tubular damages caused by myeloglobinuria. Detection of IgM antibody indicates recent infection, as it can be detected 3 to 5 days after onset. IgG antibody is detected on days 8 to 9 to 10. IgG detection is used to identify the individual recovered from the disease or to evaluate the serological prevalence in the community. Viremia develops soon upon infection, followed by the increase of IgM and IgG. Antibodies are not detected during the first few days following infection, so RT-PCR is used to test during these first few days. So far, 17 bat species have evidence of filovirus infection. Antibodies of Marburg, Zaire, and Western Ebola and Bombali were identified in bats. Antibodies for from Bundibugyo, Sudan, and Thai forest Ebola virus were not identified in bats. Countries in red had Zaire Ebola virus infection. Those in pink had confirmed antibody positive. As you can see, there is a close relationship between the habitat of bat with confirmed Zaire Ebola virus antibody and the countries with outbreak. In 2004, when there were large outbreaks of acute respiratory symptoms and miscarriages in the two big farms in the Philippines, porcine reproductive and respiratory syndrome virus, porcine circovirus 2, and Western Ebola virus were detected from the pig in the farm. During the investigation of Western Ebola virus in bat in China and in the Philippines in 2011, it suggested that the bat of the regions were the natural host of Western Ebola virus. Viral gene and antibody were detected from the bat in the Philippines. Western Ebola virus gene was detected from the spleen of the pig detected and died of porcine reproductive and respiratory syndrome virus in the Filipino pig farm. The chart summarizes the types of Ebola virus and pathogenicity. Zaire Ebola virus is highly pathogenic to human as well as monkey. Its natural host has not been confirmed, but the studies so far suggest fruit bats including Hispinigus monsteratus, Rosetus, Aegyptiacus, Epimopus francati, Myonecteris tolcata, and fatality is very high, between 60 to 90%. On the other hand, Western virus found in Asia is pathogenic to monkeys but not to humans. Though its natural host has not been identified, the putative host is Rosetus ampliciscaudatus. Now, let's look at the relationship between animal and Ebola hemorrhagic fever. An investigation of wildlife was conducted in 2001 when there was an Ebola outbreak in Democratic Republic of the Congo. First, Animal Mortality Monitoring Network was established soon after the human outbreak was announced for epidemiological study by Wildlife Conservation Society and World Wildlife Fund. The network team studied the dead animals in the area centering the national park covering Congo and Gabon to identify the cause of death. Between August 2001 to June 2003, 98 dead animals were found in the area of about 20,000 square kilometers. Bodies of 3 major species, namely 50 gorillas, 15 chimpanzees, and 14 duikers were collected. This is the fort of duiker, a deer-like antelope. There were 2 peaks from animal death. The first peak was between November to December 2001 in Gabon, where 51 dead bodies, including 30 large apes and 8 duikers, were found. The second peak was observed in December 2002 to February 2003. 20 bodies, including 17 large apes, 1 duiker, were found in Loshi Gorilla Sanctuary. Between October 2001 to December 2003, in the area bordering Gabon and Congo, there were 5 Ebola patients. The outbreak ended up with 330 cases with 264 deaths. Epidemiological studies suggested that bodies of at least 2 duikers, 2 chimpanzees, and 2 gorillas were involved or suspected of involvement with infection of 6 index cases. The outbreak in Congo between January 2001 to June 2002 had 30 cases and 25 deaths. 1 gorilla and 1 duiker were suggested to be involved in the infection of 2 index cases. Thus, it is suggested that the outbreak of wildlife precedes that of human. Thus, Ebola virus circulating among wildlife is transmitted to humans through contact and spread among people, leading to an outbreak. This is about pet animal and Ebola hemorrhagic fever. At the time of outbreak in Gabon between 2001 and 2002, canine antibody prevalence was investigated. 18% of dogs were Ebola virus antibody positive. These results show possible Ebola infection to dog, but no virus has been isolated. Currently, we cannot decide whether dog and other domestic pet develop infection with viral replication, nor can we decide whether they release virus in the body fluid. Since not much is known about the Ebola infection of dog and other pet animal, careful risk evaluation is needed when an owner is infected with Ebola and has close contact with the animal. If a pet animal has possible exposure to the virus, the animal needs to be closely monitored under the quarantine measures by a veterinarian for at least 21 days following the last potential exposure. Now I'd like to talk about the sensitivity of Ebola virus. We bred 8 strains of inbred and wild mice. We analyzed the sensitivity of Ebola virus using collaborative cross mice with genetic diversity. Collaborative cross mice showed phenotypes of varying severity from tolerant to lethal against mouse-adapted Ebola virus infection. 10 strains of collaborative cross mouse were screened to identify clinical, virological, and transcriptome characteristics distinguishing between lethal and intolerant phenotypes. In tolerant mice, immunological and inflammatory responses were strictly controlled. Inflammatory macrophages were reduced, and the increase of antigen-presenting cells, namely B1 cell and gamma delta T cell, was shown. On the other hand, the lethal phenotype is characterized with initial control of gene expression, lymphocytopenia, and followed by uncontrolled inflammatory signal transduction and death. With machine learning, the entire data was applied to human. In the West African Ebola patient cohort, outcome was predicted with 75% accuracy. And this is schematic of the previous result. In the sensitive mouse, Ebola virus first targets macrophage and dendritic cell. It inhibits antigen-presenting T cell activation and regulation of inflammation, inducing neutrophil degranulation and pro-inflammatory cytokine storm. In the tolerant mouse, although Ebola virus does not infect B1 cell, it induces gamma delta T cell activation through antigen presentation. Currently, many drugs for Ebola hemorrhagic fever are under development across the world. This shows the list of drugs under clinical investigation. There are monoclonal antibodies, cocktails of antibodies, and cocktails of monoclonal antibodies. Remdesivir is reported as a drug to treat COVID-19. It aims to block nucleic acid synthesis. There are various vaccines for Ebola hemorrhagic fever under development around the world. These vaccines are currently in the clinical trial. These include VSV or vesicular stomatitis of domestic animal-based vaccine and adenovirus-based vaccine. In conclusion, with regards to One Health and zoonosis, maintained health of animal and environment prevents zoonosis and maintains human health. Maintained human health also maintains animal and environmental health, and that is the objective of One Health. Thank you for your kind attention.